used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living.
A novel gene editing system known as CRISPR-Cas9 may allow us to modify, delete, or correct specific areas of our DNA in order to treat these diseases. The system is comprised of two parts: Cas9, an enzyme that cuts DNA, and a guide RNA whose sequence directs Cas9 to a specific location in the DNA where the edit should be made. Cas9 associates with the guide RNA to form a complex that can be easily and precisely targeted to a desired site in the DNA. The CRISPR-Cas9 gene editing process begins when the complex recognizes and binds to a short segment of DNA adjacent to the target site. This initiates unwinding of the DNA helix, which allows the guide RNA to pair with a specific target sequence in the DNA. If the sequences pair precisely, Cas9 cuts the DNA, forming a double-strand break. Our cells respond to such breaks by activating natural DNA repair pathways.